Let us study today about the enzymes after the revisions. Let us look at the various points about the enzymes. Enzymes are biological catalysts. Please remember that they are catalysts. They increase the rate of reaction, but they are generally used into the biological or biochemical processes, and that is why they are considered as a biological catalyst. They are catalysts, but they are used into the biochemical processes, and that is why they are considered as a uh, biochemical or biological catalyst. When it comes to the various kinds of reactions, let us try to study them. Uh, uh, Almost all the enzymes are globular proteins. So this is a very important characteristics that we study, that all the enzymes are uh, globular proteins. So these enzymes are globular proteins. They are very specific for a particular reactions or particular substrate. So they work on only particular reactions, or they work on particular substrates only. They do not work for all the reactions, and they do not work from all the substrates. Next, they are named after compound or class of compounds. Their names are given after the compound or the class of the compounds which they act successfully. So they actually they are named after compound or class of compound which they act on successfully. Sometimes they named after the reactions. So sometimes they are named after a particular reaction which they catalyze, and their names generally ends with the suffix ase. For example, oxidoreductase. For example, oxidoreductase in some biochemical reactions, one enzyme oxidize the another enzyme. One enzyme oxidoreductase in some biochemical reactions, one enzyme oxidize one substance, and the same enzyme is used to reduce the another substance. This enzyme is called oxidoreductase. So here the enzyme for one reaction it oxidizes the substances, for another reaction it reduces the substances. Such enzymes are known as the oxidoreductase. This way the name of the enzyme can be based on the type of reactivity it shows. This way the name of the enzyme can also be the type of reactivity which this enzyme shows, my dear students. Mechanism of enzyme action. We have already studied into the surface chemistry. We have studied it as the lock and key model, if you can remember. In the surface chemistry, lock and key model we have studied. So we have studied about the mechanism of this enzyme catalysis as a lock and key model. Enzyme is called the biological catalyst, so it reduces the activation energy for the reaction and cause reaction to take place faster. As discussed in the surface chemistry, enzymes follow the lock and key model, and one enzyme is used for specific substrate only. So let us understand this specific reaction with some of the examples. Hydrolysis of sucrose, if done without enzyme sucrase, activation energy is 6.22 kilojoule per mole. But if it means sucrose hydrolysis is done in the presence of the enzyme sucrase, the activation energy decreased and it becomes 2.15 kilojoule per mole. So enzyme decreases the activation energy. Please remember that the mechanism of enzyme is even based on the decrease into the activation energy, and thus these enzymes also decreases the energy, and this way we get the enzyme, they catalyze the various reactions. Now my dear students, we want to study about the vitamins. So let us move forward. If there is anything which you could not understand about enzymes, please let me know. बच्चों अगर इसमें कुछ समझ में नहीं आया एंजाइम के बारे में तो पूछ सकते हैं आप. ऐसा कुछ है जो आपको एंजाइम के बारे में समझ ना आया हो तो प्लीज पूछ लीजिए. एटलीस्ट बता दीजिए सब समझ में आया है.
No doubt, sir.
Yes.
Thank you, dear students. Now let's move to the vitamins. Vitamins, some organic compounds are required in the diet in a small amount. If they are not consumed, their deficiency can cause diseases. These organic compounds are needed for the normal maintenance of the health and for the optimum growth of the living organism. Please remember these two words: normal maintenance of the growth, normal maintenance of the health, and for the optimum growth of living organism. They are vital, and they are called the vitamins. Vital means necessary. They are very much needed, and that is why they are considered as a vital, and they are called vitamins. My dear students, let me tell you once again that vitamins are the substances, or let us understand the definition so that we can understand it well. Vitamins are the organic compounds required in the diet. First thing is they should be required in the diet in a small amount. They should be required in a small amount to perform specific biological functions. They are required to perform the specific biological functions, and why they are needed for the normal maintenance of health and optimum growth. For the normal maintenance of health, HEALTH. This health should be written for the normal maintenance of and optimum growth of the and health of the organism are called vitamins. So when you write the definition of vitamin, my dear students, you need to remember that it is needed for the normal maintenance of health. It is needed for the optimum growth by the organism, and they are needed in a very small quantity in our diet, but they are essential for diet, so they should be included in the diet, and they are useful for the to perform the biological function. So if you remember these 4 points, you can easily define. Let me read the definition once again. Organic compounds required in the diet in small amount to perform specific biological functions for the normal maintenance of health and optimum growth of the organism are called vitamins. So this is how, my dear students, we can understand the definition of the vitamins. Vitamins are given name according to their discoveries. As they are discovered, their names are formed as A, B, C, and so on. So vitamins, we have A, vitamin V, B, vitamin C, vitamin D, vitamin E, vitamin H, vitamin K. These are some of the examples of the vitamins that we have. The structure of vitamins are not the same, or all the vitamins do not have similar functional groups, but initially discovered vitamins have amine group present in them. So initially, whatever the vitamins they were discovered at that time, these all vitamins generally had a amine group present in them, and that is why thus they called vital amines. Vital means necessary, amines means having the functional group amine, and thus they are called the vital amines, and slowly these vital amines are named as vitamins. But later on, it is found that all vitamins do not have NH2 or amino group. So last E, this E from the vitamins, from the amine is dropped, and now they are known as the vitamins where there is no E after N. So this is how the name vitamin came into the existence, my dear students. Classification of vitamins. Vitamins are classified on the basis of their solubility in water or fat. So there are two classes of vitamins. One is a water soluble vitamin and another one is fat soluble vitamin. Let us study first about the fat soluble vitamins. Vitamins A, D, E, K are called the fat soluble vitamins and are stored in a liver as well as in the adipose tissue. Now, what is adipose tissue? Adipose tissue is the fat storing tissue available in our body. So these vitamins, vitamin A, D, E, K, are called the fat soluble vitamins. They are stored in our liver or they are stored into the adipose tissues. Then the second class of vitamin we have, which are known as the water soluble vitamins. Vitamin B and vitamin C are water soluble vitamins. Vitamin B is not a single vitamin, but it is the class of vitamins or group of vitamins. That is why they are called vitamin B group or vitamins, or they are also called vitamin B complex. So they are called B group vitamins or they are called the vitamin complex. So vitamin B is not actually a single vitamin, but it's a group of vitamins. We have even seen that our vitamin B and vitamin C are the water soluble vitamins. Water soluble vitamins are dissolved in water and they are excreted. Excreted means thrown out of the body. So they are excreted from our body through the urine except vitamin B12. So except vitamin B12, all the vitamin B and all the vitamin C can be thrown out from our body or excreted from our body through urine. Thus, they cannot be stored in our body. So, vitamin B and vitamin C, we cannot store them in our body. We should take these vitamins regularly in our food. We should consume these vitamins in our diet regularly. We have also vitamin H is also the water soluble vitamin known as biotin. So we even have water soluble vitamin names as vitamin H, and it is the another name of vitamin H is biotin. This is the extra information, but it is better if you know it. Now, my dear students, we want to study one definition of one disease, that is what it is known as the convulsions. It is a condition in which our body shivers and we call it fit. Due to this, our contracts and relax rapidly and repeatedly in an uncontrolled shaking that causes the uncontrolled shaking of the body. Once again, convulsions, it is a condition in which our body shivers and we call it fit. Due to this, means due to fit or due to the convulsion, our muscle contracts and relax rapidly and repeatedly in uncontrolled, and that is what it causes the uncontrolled shaking in the body. Now, my dear students, let us study the various vitamins, their source, and what are the diseases or deficiency diseases. So, deficiency diseases, these are the source and these are the various vitamins. So, first we have vitamin A, which is also known as the retinol. It is obtained from the fish, cod liver oil or fish liver oil, carrots, butter and milk. And the disease due to this happens is known as the xerophthalmia. That is what it is known as the hardening of cornea of eye, or it may also cause the night blindness. Then we have vitamin B1, which is known as the thiamine. Vitamin B1 or thiamine can be obtained from the cereals, yeast, milk, green vegetables. And the disease which are caused by these are the beriberi, loss of appetite, and retarded growth. Then, my dear students, we have vitamin B2, which is also known as the riboflavin. This vitamin is obtained from the milk, egg white, from the livers and from the kidneys. And if it is not available in our body, it causes the diseases like chelosis, that is fissuring or fissuring at the corners of the mouth and lips, or they can cause the digestive disorders and they can cause the burning sensations. Then, my dear students, we have vitamin B6, which is also known as a pyridoxine. This vitamin B6 can be obtained from the yeast, milk, or egg yolk. They are also obtained from the cereals, and they cause vitamin B6 cause the convulsions. Then, my dear students, we have vitamin B12. Vitamin B12 can be obtained from the meat, fish, egg, and curd. It causes the pernicious anemia, that is the RBC deficiency in the hemoglobin. So, this is what it is known as the pernicious anemia, that is the RBC deficiency in the hemoglobin. That is what it produces due to the vitamin B12. Then, my dear students, we have vitamin C. This vitamin C is also known as ascorbic acid. These are vitamin C can be obtained from the citrus fruits, amlas, and green leafy vegetables. If there is a deficiency of vitamin C, the disease caused is known as the scurvy or bleeding gums. Then, my dear students, we have vitamin D. This vitamin D can be obtained from the exposure to the sunlight. Vitamin D can be obtained from the fishes, or vitamin D can be obtained from the egg yolks. The deficiency of vitamin D causes rickets, that is the bone deformity into the children, and osteomalacia, that is the soft bones and joint into the adults. So vitamin D causes rickets, which is generally seen into the children or kids, and this osteomalacia is generally seen into the adults. Then, my dear students, we have vitamin E. Vitamin E can be obtained from the various vegetable oils like wheat germ oil or sunflower oil, etc. And it causes the increased fragility of RBCs and muscular weakness. Fragility means they can be easily broken or they can be easily ruptured. So thus the RBC can be easily ruptured in the deficiency of vitamin E, and it also causes the muscular weakness. Then we have vitamin K. Vitamin K can be obtained from the green leafy vegetables, and if you do not, or if you have deficiency of vitamin K, it causes the increase in the blood clotting. So blood clotting time is being increased by the deficiency of vitamin K. These all, my dear students, we have studied. It's an extra page that I have given you. The last thing what I have not been discussed is this vitamin H. This vitamin H is also known as the biotin, and biotin can be synthesized by the intestinal bacteria or yeast, or it can be also obtained from the vegetables, and it can also be obtained from the fruits. The deficiency of vitamin H can cause the skin lesions, or it can cause the loss of appetite. It can it can also cause the weakness, and it can also cause hair fall. It can also cause paralysis in these severe conditions. So these are the various vitamins, my dear students, we have studied. Now we want to study about the nucleic acids. So let's study about the nucleic acids, my dear students. Nucleic acid is formed in the nucleus of cell, especially in the chromosomes. So nucleic acid is formed into the nucleus of cells, especially into the chromosomes. In the chromosomes, we find DNA. DNA is the deoxyribonucleic acid, and RNA is the ribonucleic acid. These are made up of units called the nucleotides. Nucleotides are made up of nucleosides. Nucleosides are made up of sugar and base. So this is the order that we understand, that we have RNA, DNA, then nucleotides, and then nucleosides, and then we have sugar and base. So this is how it is, the order of the going from bigger to the smaller. Sugars are of two type in this. Either we have D-ribose sugar or 2-deoxyribose sugar. You can see here, my dear students, that we have beta D ribose, and here we have beta D2-deoxyribose. So on the second carbon, you can see that both are hydrogen here. Instead of OH, the oxygen has been removed, and that is why it is known as the deoxyribose sugar. This is a ribose sugar. So this is how we understand the structure of the two sugars. Now, apart from sugar, we have bases. The two bases are purine and pyrimidine bases. So let us understand about purine. You can see the structure of the purine bases, which are known as the adenine and guanine. We can even study about the pyrimidine bases. They can be cytosine, thiamine, or uracil. This uracil can be seen only into the RNAs. So uracil cannot be seen into the DNAs. Now combination of base and sugar is called nucleoside, in which pentose sugar, if bonded with first carbon with the base. So this is the first carbon, my dear students, and with this first carbon, this pentose sugar is bonded with the base, and the structure you can see is called the nucleoside. This is the nucleoside of RNA. This is the nucleoside of DNA. You can see here on the second carbon there is no OH, and that is why it is deoxyribosugar, and that is why it is DNA. You can easily understand that nucleoside is equal to base plus sugar. But now nucleotide is equal to base plus sugar plus phosphate group. So you can see here that in this figure, you can see the nucleotide I have shown you. This is your nucleotide, my dear students. Here you have this sugar. This is your base. And here you can see your phosphate group. This is what it is, the phosphate group. So this phosphate group even include this part. I'm sorry, this phosphate group includes this part even. So this is the phosphate group, which is a PO4 minus 3. So this is the phosphate group. Now you can see that from first carbon, the sugar is bonded to base, whereas this is second, this is third, this is fourth. So from the fourth carbon, it has been bonded to the phosphate group, and this is how we get the nucleotide. Nucleotide has three things: base, sugar, and phosphate group. You can easily understand that when the nucleotide is involved into the nucleic acid, the another nucleoside is combined with the third carbon of the nucleotide. Please remember that if we have the adenine and thymine, this adenine and thymine combine with each other with the 3 hydrogen bond, and cytosine and guanine, they combine with each other into the 2 hydrogen bond. This is what you need to remember. Base placed are different in sequence, but their sequence is called the genetic code, which is important for the maintenance of cell or for the cell formation. So here you can see, my dear students, these are the various bases I have shown you. Let us revise these structures again. We have seen that we have two bases, purine bases and pyrimidine bases. Pyrimidine base are cytosine, thymine, and uracil. Cytosine and thymine are found in DNA, while cytosine and uracil are found in RNA. Now you can see we have various structures of bases. This is uracil, this is cytosine, this is thymine. Purine base are adenine and guanine. You can easily see them. There will be hydrogen bonding among these two strands of DNA, and there will be double hydrogen bond between A and T, while the triple hydrogen bond between C and G. That is what you can easily understand. Now you can see here the exact structure of either DNA or RNA. You can see here that we have two strands. One strand is going into this direction. You can see here, it is very easy for you to understand. Please look at this, my dear students. See, this is going in the upward direction. This is going in the downward direction. These are the two strands that are found into the DNA. That is why we call it double helical structures. In this double helical structure, you can see that this portion is known as a nucleotide. In this nucleotide, we have sugar, we have base, and this is what it is, the phosphate part. You can see that the phosphate part is present on the fifth carbon, and this from the fifth carbon, it is bonded to the third carbon of the previous group. This is the fifth carbon, actually it is found here, and this from the fifth carbon, it is bonded to the previous nucleotide. The bond between two nucleotides is known as the phosphodiester linkage, and by the phosphodiester linkage, two nucleotides can be connected. Now these two trends, one trend is going upward, another is going downward. It is also the trend or the strand that is made up of or helical that is made up of various nucleotides. How these two different helicals can be bonded with each other? They are bonded with the hydrogen bonding. You can see that there is a double bond between the hydrogen, there is two hydrogen bonding between the adenine and thiamine, but there are three double bonding between the cytosine and guanine. So this is how the two helicals are bonded with the hydrogen bonding, whereas two nucleotides are bonded with the phosphodiester bond. And in each case, when we have sugar, the sugar having on first carbon, the sugar is having base, and on fifth carbon, sugar is bonded with the phosphate group, my dear students. The same thing, if we really repeat, we will understand it well. Let us have one more look to it. You can see, my dear students, that when we have a cell, the cell has a nucleus, nucleus has a chromosome, chromosomes has a DNA, DNA is made up of the nucleic acid. Now we have nucleic acid, or DNA is a nucleic acid has got phosphate, sugar, base, which is known as the nucleotide. If the phosphate group is not present, these nucleotides are bonded with the phosphodiester bond. When we have nucleoside, only sugar and base are present, and we have various sugars, whether we have beta D ribose sugar or beta D oxyribose sugars, and then apart from sugars, we have bases. These bases are purine bases and pyrimidine bases. Purine bases are adenine and guanine, whereas pyrimidine bases are thiamine and cytosine. Apart from that, the pyrimidine base is uracil, which is found only into the RNA. You can even see the figure here, that in this figure you can easily see that we have a nucleotide which is having sugar. You can see that in a nucleotide we have in this nucleotide we can easily see, my dear students, that we have just a minute. My goodness. In this figure, my dear students, you can easily see that this nucleotide has got, this is the base, this is the sugar. This sugar is from first carbon bonded to base, and from fifth carbon it is bonded to the phosphate group, and from third carbon this nucleotide is bonded to the another nucleotide, and this is what it is known as the phosphodiester bond. We can even see that base are bonded to the another strand by either triple hydrogen bonding or double hydrogen bonding. So this was a good revision, my dear students. Now you can again see the same things here. A simplified version of nucleic acid chain is as shown here. You have sugar which is bonded with base, then you have phosphate, then again you have sugar, base, phosphate, and these are the N. So that is what it causes the nucleic acid. You can see even the figure of DNA, where this is the double helical structure of DNA, where guanine and cytosine are bonded with the triple bond, and adenine and thiamine are bonded with the double bond. And these are the two different strands you can see. So this is how we understand the structure very easily and very nicely, my dear students. Information regarding the sequence of nucleotide in the chain of a nucleic acid is called the primary structure. Nucleic acids have secondary structures also. James Watson and Francis Crick have a double strand helical structure, or they have developed, or they explain the double helical structure of the DNA, my dear students. Let us study further. Watts, Crick and Jemsons, they have given us the double strand helical structure for DNA. Two nucleic acid chains are wound around about each other and held together by the hydrogen bonds between pairs of bases. These two strands are complementary to each other because because the hydrogen bond are formed between the specific pairs of bases. Adenine forms hydrogen bond with the thiamine, whereas cytosine forms hydrogen bond with the guanine in case of DNA, my dear students. The secondary structure of RNA, helices are present, which are only single stranded. So in RNA we have only single stranded structures. Sometimes they fold back on themselves to form double helical structures. RNA molecules are of three types and they perform the different functions. Please remember, RNA molecules are of three types and they perform different functions. They are named as messenger RNA, that is mRNA, ribosomal RNA, that is rRNA and transfer RNA, that is tRNA. So this is how, my dear students, we study about the various types of RNAs. Now we want to study a little bit more about the genetics or genetic part of this chapter. There was a scientist, his name was Hargobind Khurana. Hargobind Khurana was born in 1922 and he obtained his MSc degree from the Punjab University, Lahore. He worked with the professor Valdemir, Valdemir Prelog, who molded Khurana's thoughts and philosophy towards the science, work and effort. After a brief stay in India in 1949, Khurana went back to England and worked with the Professor G.W. Canner and Professor E.R. Todd. It was at Cambridge, UK, that he got interested in both the proteins and nucleic acids. Dr. Khurana shared the Nobel Prize for the medicine and physiology in 1968 with Marshall Neinberg and Robert Halley for cracking the genetic code. So this genetic code, that is the AGCT code, that is the code that is made up of adenine, guanine, thymine and cytosine, AGCT code, which was cracked by our Dr. Hargobind Khurana, and for that he was awarded the Nobel Prize too. So that was about Hargobind Khurana. Now we want to study about the special method developed by our Hargobind Khurana by cracking the genetic code, which is known as the DNA printing. It is known that every individual has a unique fingerprints. This occurs at the tips of the fingers. So as we have unique fingerprints, same way there is something that we are going to study and have been used for identification of a long time, but this can be altered by surgery. So these fingertips are using or are being used for identification of a person, but these fingertips can also be altered by a small surgery. A sequence of a sequence of bases on DNA is also unique for a person. So each person has a unique sequence of DNA which is made up of AGCT and information regarding this called DNA fingerprinting. So the arrangement of adenine, guanine, thymine and cytosine, AGCT into the DNA, this code is unique for each and every human being, and on the basis of this code we can identify the person. It is same for every cell and cannot be altered by any known treatment. So there is no treatment available by which this DNA coding can be altered. And this is what it is known as DNA fingerprinting. And nowadays, my dear students, this DNA fingerprinting is used in forensic laboratory for identification of a criminals. So if criminal has done some crime and has ran away, and if you want to find out from his blood cell or from his hair cell or from any of his or her cell, we can identify whether the cell belong to a criminal or not. To determine paternity of an individual, suppose if a child, if more than one person say that they are the parent of a particular child, then by doing a DNA test, we can easily identify which one is the true parent of a particular child, because this unique code also can be related to the parents. The identity of the dead bodies in any accident by comparing DNA with the parents of children. So if a person or a human being dies into the accident and it is difficult to identify that person, then looking into the DNA code of that person and matching them with their children or their parents, we can identify whether the person belong to that parent or child or not. And the last one is to identify the racial group to rewrite the biological evolution. So my dear students, during the process of writing the biological evolution, which species belong to what kind of groups or what kind of family, if we want to determine it by using the genetic code of that species, we can rewrite the racial groups into the process of the biological evolution. And then my dear students, uh, biological function of nucleic acid. What are the biological functions of the nucleic acid? DNA is a chemical basis of heredity and may be regarded as the reserve of genetic information. So DNA is the reserve of the genetic information. It takes the information of genetics from uh, one generation to the another. DNA is exclusively responsible for the maintaining the identity of different species of organism over a million of years. So if you look into the species for million years, or you can see for many generation, the DNA of same species remains the same, and it maintain the code in a different manner. A DNA molecule is capable of self duplication during the cell division, and identical DNA strands are transferred to the daughter cells. So DNA itself can duplicate itself, and the same kind of DNA can be transferred to the new generated cell called the daughter cells. Another important function of nucleic acid is the protein synthesis. The another important function of nucleic acid is protein synthesis. It is used into the synthesis of protein, the cell synthesis in the cell. So in the cell, whenever the protein is being generated, for that the coding is needed, and this coding is generally in the form of the DNA code. Actually, the proteins are synthesized by the various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in DNA. So what happens, my dear students? The chromosome produces the DNA code. This DNA code is transferred to the cell portion by the messenger RNA, and this is how the message goes, and then the protein is synthesized by the various RNA and the various organelles present into the cell. So this is how, my dear students, the initiation of the protein synthesis in the form of code is done into the chromosome, and by using the DNA. Even the DNA is responsible for the hereditary transfer of the characteristics, and DNA is very important for we people to understand. It is also used into solving of the various criminal cases, as well as for the rewriting of the biological evolution, at the same time for identification of the criminals or parents, or paternity of an individual. So this is how, my dear students, we complete or finishes the discussion about the nucleic acids, nucleotides. And if you have any questions now, you can ask me. बच्चों, आप मुझे सुन पा रहे हो?
हाँ सर।
अगर आपको कुछ पूछना है, अगर इसमें कुछ समझ में नहीं आया तो आप पूछ सकते हैं, या फिर हम रिवीजन की ओर चलते हैं. डू यू हैव एनीथिंग टू आस्क?
नो सर.
नो सर.
ऐसा कुछ है जो आपको समझ में नहीं आया? Okay then, बच्चों, have a nice time. Let us do the required revision, and